Hello, everyone. Welcome to today's live broadcast, The Art of War, Unleashing the Immune System to Fight Cancer. I am Brenda Kelly-Kim of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Affymetrix. Affymetrix, a part of Thermo Fisher Scientific, is a pioneer in microarray technology and a leader in genomics analysis. Affymetrix develops and provides innovative technology that will enable multiplex and parallel analysis of biological systems at the cell, protein, and gene level, facilitating the rapid translation of results into biology for a better world. Our extensive portfolio of translational and clinical solutions enables scientists and clinicians to rapidly translate their research into understanding underlying disease mechanisms, identifying biomarkers for personalized medicine, creating novel molecular diagnostic tests, and improving genetic marker-assisted breeding programs in agriculture for human health and wellness. We strive to provide leadership and support, partnering with our customers in their quest to translate biological knowledge into clinical application and routine use. We are proud to be part of the Thermo Fisher Scientific family, helping to advance our mission to enable our customers to make the world healthier, cleaner, and safer. For more information, please visit www.affymetrix.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many of your questions as we can today. You can enlarge the slide window by clicking on the screen icon at the lower right of the presentation window. If you have any problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Dr. John Connolly. Dr. Connolly is a senior principal investigator and director for translational immunology at the Institute of Molecular and Cellular Biology. Additionally, Dr. Connolly serves as program director for the A-STAR program in translational research in infectious disease, a multidisciplinary center focused on vaccine development. As a human immunologist, his research focuses on immunotherapy for cancer and target discovery for immune modulation. Dr. Connolly is adjunct associate professor of immunology at Baylor University. He received his PhD in immunology from Dartmouth Medical School and studied human dendritic cell biology under Dr. Michael Fanger. During his time, he was involved in the development of immunotherapeutic preclinical models and clinical trials for glioblastoma multiforme. He moved to the Baylor Institute for Immunology Research, a fully translational research institute dedicated to rationally designed biologics, vaccines, and cell therapy for cancer. Dr. Connolly served as the director of research initiatives for the Baylor Research Institute, a large integrated translational research resource and multi-institutional programs that involved a number of international sites. During his tenure at Baylor, Dr. Connolly was the Central Core Facility Director of the NIAID Centers for Translational Research on Human Immunology and Biodefense, an NIH-funded consortium of basic translational research and clinical trials focused on vaccine design. Dr. Connolly is the past president of the board of directors of the American Cancer Society in North Texas and founding director of the Singapore Immunology Network. I will now turn it over to Dr. Connolly for his presentation.
Okay, great. Thank you very much for that introduction, and thank you all for being here today. Good morning to everyone in the U.S. and good evening to all my colleagues here in Asia. So my name is John Connolly, as you just heard, and it's really quite an opportunity for me to talk to you guys today. I'm really excited to speak about our work in immunotherapy for cancer, and I'd like to, during the next hour or so, highlight some of the work that we've done in early on, both when we were in New Hampshire and in Texas and then here in Singapore, and talk a little bit about some of the fundamental research that went into the design of some of our immunotherapies, but also to highlight some of the really important fundamental discoveries that are being made throughout the field of immunotherapy for cancer, and some of the fantastic work that's being done by other labs in spaces like biologics. And hopefully by the end of this, you'll have a context in which not just to understand these results, but also in which to sort of understand how we're moving forward, what the biggest challenges are here in this field of cancer immunotherapy, and how we can begin to use the immune system to fight cancer. So I think that this is an area I think you're all aware of that's really picked up enormous amounts of speed in the last 5 or 6 years. And it's been both in the scientific press as well as in the popular press as a really important breakthrough in fields of cancer, is the application of the immune system to attacking tumors. And this is based really on just wildly successful results, some incredible breakthroughs, paradigm-shifting breakthroughs. But these discoveries and this medical application is really based on decades of research that has been done, not directed at cancer, but really directed at trying to fundamentally understand what is happening in the immune system, how the immune system functions in terms of trying to direct responses to invading pathogens. And so those fundamental discoveries have really led to our understanding and application toward the fields of medicine. So just stepping back just for a second to talk about how we approach things. So my group, so as we heard from the introduction, my lab studies fundamental human immunology, and I had the good fortune of working with some spectacular human immunologists throughout my training. And what they really drilled in is really to understand the human immune system. You really have to study humans. And so to do that, we really analyze a lot of ongoing clinical trials, both our own investigator-initiated trials, as well as large phase 1, 2, and 3 clinical trials, to try to get a better understanding of the human immune response during the course of disease and therapy. And so in essence, this is really a truly experimental medicine that we're working with. And to approach this, we put together a program in translation immunology that tries to address the gaps between laboratory medicine and clinical practice. And so the first important areas, we look at clinical support, so trying to create relational databases to understand our clinical data, to organize it, research nurse staff to try to handle sample acquisition. Biobanking capability is critical, so being able to put together large cohorts of human samples in order to get high quality data from these cohorts, and we've built quite an extensive biobank in these trials. And then I think an important part of this as well is the application of technology, advanced technology. And so in this, we put together an immunomonitoring platform. An immunomonitoring platform is really an assortment of cell-based core facilities, technology-focused cores, and a common IT infrastructure that can integrate that data together. So these 3 pillars really form the crux of the program that gives us real information about mechanisms of action. So actually, how are these therapies working in the individuals they work for? And probably more importantly, how do they fail? What is the mechanism of resistance to immunotherapy? And this is something we spend an inordinate amount of time talking about and understanding, with the eventual hope, and actually in some cases we've actually done it, of understanding this mechanism and then rationally redesigning our therapy to make the immunotherapy more effective across a broader spectrum of individuals. So this is really kind of the greater vision and how we've approached things since day one in the studies. So let's take another step back to talk about the application again of these fundamental discoveries to cancer. And to do that, we really want to get a better understanding of how science in general is driven. And I think that if someone can come up with more, then that would be great. But in my mind, I think we really did some by 3 different things. So the first is luck, which I equate with hard work. And so individuals that are working and doing the same thing over and over again, you have a certain level of expectation in your experimental studies. And when something comes up different, you really go back and investigate that. And so this is really where you make some very important discoveries. This is exemplified by the discovery of penicillin, where you really see these differences, and this teaches you something completely different about what we do. The other is genius, and in this sense, There are some people who are just born, there's something wrong with them, so they just see the universe differently. And in doing so, science leaps forward with their revised understanding. And so these are of course people like Darwin and Einstein and Paul Dirac, where they see these kind of differences and it revolutionizes things. But the main driver of scientific advance, and certainly the one that I think most applies to the work that we'll be talking about today, is the advent of new technology. So a telescope is invented and Galileo can study the moons. Cook has a microscope and we have a germ theory of disease. So these kind of advances and the acceleration of technology in the past 50 years, and particularly in the past 20 years, has really driven a lot of these discoveries and given us a much better understanding of what is fundamentally an extremely complex system, that is the immune system. So with these discoveries, though, I think it's natural, and it's certainly natural for any human immunologist, to try to then drive medical advance. So science does, in turn, drive medicine, and that these discoveries are applied to, hopefully, improving the overall human condition. And so this is certainly true in the context of the immune response, where we have numerous examples where our understanding of the immune system, or at least the application of the immune system, whether we understand it or not, has led to really wonderful advances. So one of the first understandings that we have of the immune system, when you think about it, is we've divided into an adaptive immune response and an innate and adaptive immune response. So the innate immune response is really sort of the physical barriers and the quickly reactive cells like neutrophils, monocytes, the non-antigen specific responses that happen in the rapid physical defense. And these really make up the bulk of most of your protective capability. The other response that I think that most people would equate with proper immune function is this concept of the adaptive immune response. And this is typified by immunological memory. And so what this is really when an organism invades and your microorganisms attacks you, you have a specialized select memory cells that are selected for and expanded. This leads to a greater preponderance of these specialized cells in a secondary infection, so which basically looks like memory, so that you're sort of reactive to this secondary response. And so this we, of course, call the adaptive immune response, because the immune system is essentially prepared for the secondary attack. Now, although we all think of this as clearly a normal function of the immune system and something that we all understand, realistically, it's not actually normal. It's not something that we see throughout life. It's really only jawed vertebrates and very few other organisms. There's some wonderful work from Max Cooper and others looking at lamprey eels that have convergently evolved this. But realistically, it's just us, the jawed vertebrates, that actually have developed this adaptive immune response. But despite not really even understanding how this functioned for many years, we were still able to leverage the potency and power of this adaptive immune response to treat human disease. So very early on, it was identified that Athenian plague victims, even in ancient Greece, who survived the plague, could then, without risk to themselves, then treat existing patients. And so we now know that this is the product of this immunological memory that's giving them protection. And some years later, we had the work of Edward Jenner, whose observations of milkmaids who had been exposed to cowpox were then protected against smallpox. And Jenner's famous experiments, taking a boy and actually infecting him with vaccinia virus, the cowpox, and then exposing them to smallpox, demonstrated very clearly that this is mechanistically how you are actually protected, and that there's this cross protection between these two viral strains. Obviously, something that we wouldn't do today and wouldn't classify as experimental medicine, but it was certainly quite definitive. So this, even though we at the time we didn't have a very good theoretical framework in which to understand the immune system, we were still applying it. And this has led to what was inarguably the greatest success of immunology, certainly vaccinology, which is the development of vaccines. It leverages this adaptive immune response to really treat human maladies. And if you look here, there's a number of figures that are out there like this. All of them are equally impressive. When you look at the role of vaccines in protecting us against what is the scourge of the human race in the context of infectious disease, in a single century, we essentially greatly eliminated these things from problems. And this is really the development of obviously vaccines. And it culminates with probably one of the greatest advances in all of medicine, which is the eradication of smallpox in the late 1970s. So this is fantastic. Even without understanding the adaptive immune system, we're still able to leverage it to develop these vaccines. But in order to rationally design therapies for something as difficult as cancer, we needed a theoretical framework in which to understand it. And so very early on, it was recognized that they are specialized cells within the immune system, that they're dynamic, that they communicate with each other, they polarize. There's a kinetic nature to these cells. I already mentioned that there's immunological memory, something extremely complex. And so how do we start? How do we approach this so that we can then experimentally address this and begin to manipulate and utilize it to treat cancer? Well, I think that in the past 100 years, I think one of the greatest postulates that sort of came forward in which a framework to understand this was really exemplified by this statement here, which is the inscription on the temple of Apollo in Delphi. And for those of you who can't read Greek, it says know thyself. And so that concept, that statement of know thyself, really is a framework in which we've understood immunology in the past 100 years. It's a fundamental theorem of immunology. And this was best verbalized by a professor at the College of France, Claude Bernard, famous scientist in his own right for his research, a vivisectionist, an anatomist, a philosopher, a poet, and a real proponent of the scientific method. It was really his philosophy and the philosophy at the time that led to his statements around that, in contradiction to an animal which is in humoral balance with the universe, really the primacy of the organism is essential to our understanding of reactions with nature. So he was a man of his time in the sense that he was a humanist. It was man against nature. Self versus non-self became this context in which he was describing his contemporaries' experiments in immunology. Although this was a fantastic postulate, it lacked evidence. So, although it was more of a theoretical framework that was in keeping with the Industrial Revolution and its the man of his time in the period. And so the theoretical construct in which to understand immunology was this concept of protected self serves as the central theme, that an entity is identified as self is protected actively, and then identified as non-self is actively attacked. And so that really became this understanding of how the immune system could possibly function. Although it was used and understood to look at the work of Pasteur and Ehrlich and many of these other people, it really wasn't until like the 1950s that we began to actually see molecular evidence of this actually holding up. And this came from the work of transplant surgeons who were trying to understand why transplanted organs were accepted versus not accepted, and in doing so, essentially identified the molecular nature of self. And so it was known that you could transplant organs between identical twins with some level of success, and that there was something about genetically ingrained that would recognize as self. And that by the 1950s, the human major histocompatibility antigens, or the MHC molecules, were serologically identified. These molecules in all of us are the most diverse regions of the genome. And because of this, they really serve as, in essence, molecular signatures of self. Okay, so this is fantastic, and it supports the overall hypothesis of self versus non-self being how the immune system functions. And I should say that throughout this time, there are competing hypotheses that were out there, but this one has stood the test of time, particularly when we began to investigate the questions of non-self. So we all know, there are many cases in which the immune system doesn't react to the lack of self, and therefore it was hypothesized that there must be, for this postulate, this theorem to be true, there must be a molecular mechanism of non-self recognition. And it took a really long time for us to begin to then recognize this. But these markers of non-self do exist in nature. And so pathogens have a number of different molecular patterns, so things that they really can't change. They're like themselves, like viruses, like bacteria, but completely unlike everything else that's in humanity. And we call these patterns molecular pathogen-associated molecular patterns, or PAMPs. And so these are signatures these are proteins, or these are lipids or molecules that are very difficult for the organism to change. and they're very well conserved across a broad category of species, but there's something that they can't change. And so we're able to then recognize these, and they represent invariant or minimally variant markers of non-self. So these are targets potentially for receptors to recognize, but it wasn't really until the early 1990s or early mid-1990s that these three gentlemen, along with their teams and labs and a number of other individuals, but really went out and pioneered and identified these receptors, germline encoded receptors that can recognize non-self molecules. And so this is the work of Charles Janeway, Bruce Beutler, and Jules Hoffman, who identified these toll-like receptor system that can recognize non-self and is one of the major non-self recognition systems that are out there right now. And so the TLR system can recognize these PAMPs, these molecular patterns. It can see virus-specific patterns. It can see bacterial-specific or fungus-specific and identify them as non-self and therefore initiate an immune response against these molecules. Importantly, it's not just the recognition of these single organisms. The coordinated expression and combined combinatorial signaling between these receptors allows you to tailor the responses against these pathogens so that you can have a diversity of specialized responses that are directed to deal with different kinds of organisms like gram-negative, gram-positive, flagellated bacteria, viruses, etc. And so this system, and it does typify a number of other non-self recognition systems, but it initiates the overall response. Importantly, the magnitude and quality of response is determined by the actual PAMPs that are expressed and the context in which they're recognized. Importantly, the combinatorial signals determine how the downstream signals are polarized. Now, this is a fantastic model. We've now sort of validated what Claude Bernard said, that we found the molecular nature of self and molecular nature of non-self, and this gives us some kind of real molecular and evidential context in which to understand overall immune responses. But one question that came about was, what are the key cells in this immune system, in this organ system, that make this decision? So do all cells do it? Are there specialized cells? And that initiate this recognition and decision of non-self to self and initiate the immune response. And so it was the work of this gentleman here, so Ralph Steinman, who in the early 1970s was working at the Rockefeller University in Zhang Cohen's lab and identified a cell type that was remarkably good in initiating and priming new immune responses, and particularly with non-self pattern recognition. And so these cells are, of course, dendritic cells. And so the cells are extremely good at initiating new primed responses and taking these molecular patterns, which are essentially innate immune recognition, and converting them and using them to instruct the development of an adaptive immune response. So quite critical cells in overall immunity. And so that's really what these dendritic cells do. So for instance, a dendritic cell can serve as a sentinel, constantly tasting and internalizing proteins or fluid or dead cell debris. And in this case, it can identify and internalize a flu virus, for instance, process that virus, and then present that virus to T cells. Now, it's not just the recognition and presentation of non-self. It is transmitting signals to instruct those T cells to become specialized antiviral T cells. And so, as we're learning it from the work of Ralph and Jacques Mongereau and Antonio Lancevecchia and many, many other famous dendritic cell biologists that are out there, exactly how this works, we're beginning to understand how we might be able to manipulate this, to generate specific kinds of T cell responses and to leverage our understanding of this dendritic cell system. So the dendritic cell, as I mentioned, sits in the periphery, so it's in your tissue, and it's constantly internalizing like a sentinel, drinking eating dead cell debris and drinking the milieu, and asking, if a cell dies, why did you die? Did you die naturally? Are you self? Or did you die due to viral infection or bacterial infection? Do you have a non-self component? And if it recognizes it, it induces this maturation signal that is qualitatively determined by the kind of pathogen it encountered. So if it's a virus signal, it polarizes it one way. If it's a bacterial signal, it polarizes it another. So the dendritic cell will then migrate to the local lymph node, where it can then interact with T cells and then instruct those T cells to become, in this case, antiviral, and then migrate out to the periphery. And the T cells, importantly, are the effectors, the guys that are actually doing the killing and destroying the infected cells. So our fundamental understanding of how to initiate these immune responses was quite a critical breakthrough, and still is. So I'll show you a movie here, just sort of demonstrating. I think with dendritic cells, it's important to sort of see what they do in order to understand. So I mentioned that they're sentinels. So I think I'll just press go somehow. Okay, so I think hopefully you guys saw what those dendritic cells were doing during the movie. They're really internalizing this, in this case, protein antigen and dead cell debris. They're constantly reaching out and constantly internalizing. And in doing so, they really are taking up a number of molecules, and they're tasting these molecules and asking, again, why did you die? So I think the next slide demonstrates the same kind of thing. But in this case, we're looking at the internalization of dead cell debris. And in this, so hopefully you'll see that in the next movie. So what you can see is that they have a huge affinity for extracellular vesicles and dead cell debris. They're constantly internalizing that. And as I said, they're diagnosing why did you die? What kind of infection led to your death? Or was this a natural apoptotic cell death, which would lead to a differential maturation program of those DCs inducing self-tolerance? And so I think in the next slide, we'll just sort of complete these movies to sort of show you what it looks like when you see non-self recognition. So these are dendritic cells, primary human dendritic cells from my blood, actually. So it's about 4 hours fresh post-blood, looking at a colony of E. coli and asking about pattern recognition. Okay, great. So I think what you guys saw there is that they got real excited by the presence of a colony of E. coli. And this is something that, that naturally happens, of course, when you see this pattern recognition. So what you didn't see, though, was during that process, the secretion of a wide variety of chemokines calling in different levels of specialized cells that would be specific for bacterial infections, so waves of neutrophils, monocytes, and polarized T cells that be attracted to deal with that infection. So directing from an adaptive immune response, directing the innate immune response. And that's not true for all dendritic cells. So here's an example of a plasmacytoid DC from the same experiment. These cells are specialized to deal with viruses and, in fact, don't react to any of the actual E. coli colony. However, I think that you can see here that upon exposure to virus, so in the absence of virus, they look basically like small round cells. Post-virus infection, this is after a few days of viral infection, you see massive dendrite sprouting. They become extremely active and internalize debris. So they look very much like activated dendritic cells. And dendritic cells, in other work, can actually communicate with each other and through these sort of microtubular structures. And our understanding of that and how it relates then to their communication, say, in the tumor microenvironment is just now being understood. Okay, so what we know is that the dendritic cells and subsets of dendritic cells can instruct T cells within the immune response. So not just diagnose the infection, the presence of non-self, but polarize that non-self, the responsive to non-self, such that you get these specialized cells that could potentially kill virus-infected cells or potentially kill tumor cells. So we know that the DCs are composed of multiple specialized subsets. They express all known TLRs. They're extremely efficient in inducing immune response, and they can distinguish between self and non-self and dictate response. So can we use them to treat disease? And so very early on during this process, in some of the most early cell therapy studies, we started thinking, can we use this very important cells to initiate anti-tumor responses? And so I guess out of all of the possible utilizations of these cells, cancer is one of the hardest and most difficult ones, because as we talked about using Claude's postulate, cancer is really a happily growing part of you. So it should be actually protected self, right? So, and this is exactly, in many cases, exactly what we see. The cancer is protected by the immune system, actively protected, and in a dominant immune tolerogenic way. And it's very difficult to generate some of these responses. And so I think when we start talking about immunotherapy for cancer, we would be remiss not to go back to really the roots of the first application of immunotherapy for cancer, which was way back during that time period when Claude and some of the earliest immunologists in the field, as I mentioned, Pasteur and Ehrlich and these guys, were just now figuring out what these immune cells did. It was really the work of William Coley in the late 1800s and early 1900s, who, after really looking at a sarcoma surgeon at the Memorial Hospital in New York City, really started looking at why people were surviving surgery or not surviving surgery, and went about and tried to find anecdotal evidence or any kind of evidence that would indicate why some people were surviving. And one of the things he noted was that there was an occasion, and on some occasions, people got post-surgical infections, which happened. It happens even now, but certainly more often during that time period, and that in at least one individual, this post-surgical infection led to what looked like a clinical remission that outlasted the actual infection itself. And so this led him to begin to hypothesize that, in fact, the immune response against the bacteria was somehow leading to a response against the tumor. And so this was quite interesting. So he began by actually infecting people with bacteria, and this led to predictably poor outcomes, so massive fever, real infections, and obviously some deaths. He then went back and really began to devise combinations of bacterial lysates and toxins and put together this Coley's toxin, which also, of course, was full of toll-like receptor agonists and stimulated non-self in the context of the tumor, led to huge fevers, real illness and sickness, and was very difficult to watch and look at, but in some cases actually did induce very impressive responses in individuals. But these responses were unpredictable, and they had terrible side effects. And so much to the horror of some of his contemporaries. And one of those contemporaries was as many of you might know, is James Ewing of Ewing sarcoma fame. So he's really a physician scientist, this guy. Very bright guy, a true champion of this new field of radiotherapy that was coming in at that time, and pulled in, with help of some of his friends and colleagues, radium into Memorial, an enormous amount of radium through his friends Douglas and mining barons and these guys. And so he brought this in and really championed radiation therapy as a new field. And this was really in contrast to immunotherapy that was going on. So this led to some difficult times between the leadership there at the Memorial, and then later on with the introduction of advanced chemotherapy, immunotherapy really kind of didn't really fit within this dominant paradigm. And so by 1955, the Coley's toxins at Memorial were stopped. That they were seen as an unproven mechanism. There wasn't solid scientific evidence. It really wasn't until many years later through Cancer Research Institute and a number of other champions, Lloyd Old and many fantastic scientists, and his daughter, Coley, are pulling together a number of studies to really look at the impact of these Coley toxins. So it really wasn't until much, much later that we saw these toll-like receptor agonists stimulating anti-tumor immunity, and it really was nearly 100 years before we saw the same levels of responses in sarcoma. So the advantage, I mentioned that, and I think it's obvious that the cancer is a happily growing part of you. So the immune system really needs a foothold to grab onto in order to see the cancer. And what it sees is really the mistakes that the cancer makes, the altered self. So the immune response is really only initiated when the cancer cell begins to make some of these mistakes. And so one of the questions that you raise is, can we build on what Coley's toxin was doing? So present these mistakes, these mutations, or these metabolic differences in the context of non-self by using stimulatory toxins like Coley's toxin. And so these mistakes that the cancer makes, some of the most important of which are called neoantigens, or simply mutations that arise as a consequence of tumor-specific events like rapid growth or p53 loss and mutagenesis. And so they're really recognized as the major target of anti-tumor immunity, particularly T-cell immunity. And so there are a number of novel therapeutic approaches that are being developed by Schreiber's group and many, many other groups that are out there to leverage these neoantigen responses. So one of the downsides about this is that these neoantigens are not universal throughout the tumor. As the tumor progresses and builds, different regions of the tumor accumulate different levels of antigens. So you might get an immune response against some of the tumor, but the rest of the tumor has different types of mutations. And so we need to really address this heterogeneity and come up with a mechanism. So one mechanism very early on that we've tried to approach is really utilizing dendritic cells to actually present these neoantigens and overexpress tumor antigens. And so in this case, this is an example of some of our melanoma clinical trials in Texas under the leadership of Jacques and Carolina, and with Joe Fay as a lead physician. We isolated dendritic cells who are isolated from blood or monocytes to derive these dendritic cells. We loaded them with cancer proteins and some of these neoantigens, and then activated them and then inject them back into the patients, and so leading to then a T-cell response within the tumor, so within the lymph node. So the T-cells now are being trained contextually that this is non-self that you're seeing to really divide and to rev up the anti-tumor immune response. They'll then migrate into the periphery where they could then destroy the tumor at the sites. And so this is some of the DC vaccines that we ran, some melanoma trial. You can see the GP100 protein in red inside the dendritic cell. So the DCs were in the vaccine itself. That is the vaccine itself. You can see it. It internalizes these and has the ability then to present these antigens to T cells and initiate strong immune responses. I think that once they're in there, they migrate not just to the draining lymph node, but you can find dendritic cells actually in the tumor sites themselves, which is good, because you'll be then taking up other tumor antigens, presumably, and cross-presenting those and expanding the breadth of anti-tumor immunity. And I think that this data typifies a lot of the research that's done by not just the Baylor Institute's team, but our team, but also the group, our own ongoing research now in dendritic cell vaccines, the fantastic research from the Ludwig Consortium, as well as a huge number of groups doing dendritic cell vaccines for a number of years. And that was that when tumors, when you see a clinical benefit, this is coincident with an immune response. But if you don't see this immune response, if you don't see the immune response, then you really don't see the clinical benefit. And this is some examples of the kind of clinical benefit that's there, that we see. And the reason I'm showing this in the next slide is really that unlike chemotherapy, you can see, when you do see a response, oftentimes the response is actually amazing and quite strong. So you see the biopsy, the subcutaneous melanoma nodules, and then biopsy-negative pools of melanin. And then in the context of this, I'm not sure if you guys can see the pointer, but in the context of the baseline chemo pre-immunotherapy, the large peritoneal mass that's pressing up against the kidney. After 8 vaccines, this is now fibrous tissue, and then large lung metastases here which are eliminated post-therapy. So one of the differences between immunotherapy and chemotherapy is really this, when you do see a response, it's a dramatic response and oftentimes very durable response. So there are a lot of dendritic cells that you can use, right? I mentioned that there are specialized cells. And so one of the dendritic cells that we can look at are some of these plasmacytoid DCs or other dendritic cells. Another is trying to take advantage of the receptors or the surfaces of DCs and actually targeting those receptors. So can we, instead of taking the cells out, can we bring the antigen there? Can we deliver both a stimulatory signal and tumor antigen? And so this is work that we did in collaboration with Medarex and Celldex to develop some of these targeting reagents. We've also worked with Gerard Zarowski and his team as well, doing spectacular targeting research. So really what this is looking at is trying to develop antigen targets, so single protein antibodies that then drag along tumor antigens. And so we can get these antigens to target dendritic cells. You can see here that in a time course they're internalized within the dendritic cell, and they are then processed and presented. They lead to strong T-cell immunity, which you can see here. This is looking at cytolytic activity against tumor targets induced by targeted cells. And so we can deliver these kind of reagents. So one key question is, what kind of cells do we want to target? And so there are a number of cell types that we can go for. One could potentially be some of these plasmacytoid dendritic cells, but also plasmacytoid DC subsets. We've demonstrated previously that there's a CD2 positive subset that appears to kill the tumor itself. And so this could be a really potentially phenomenal target to begin to deliver antigen, particularly with our better understanding of how to polarize these cells toward that Th1 immunity. And so in essence, I think the immunomonitoring that came out of these studies over our work and as well as others really came up with a few different things. One is if you don't see an immune response, you really don't, you get progression on trial. Some people, you do see an immune response, but we lose that immune response in time, which leads to disease progression and death. And then in other individuals, in a small subset of individuals on these vaccine trials, but very consistent, we see very strong immune responses that are coincident with durable response and excellent survival. And so the key really is to study all of these people, but to really go after those individuals that we can induce a response against, but it's not maintained. And to ask, what is it about this response that's fundamentally different between these individuals that can't maintain the response and those that do? So what is the mechanism of resistance to our immunotherapy? Okay, so one of the things that we identified and was really, and it's reflected in other VAX trials that were out there, is the harder we hit these individuals with dendritic cells and the more stimulatory these DCs were, the harder the immune system pushed back. So there's this reflexive pushback to sort of prevent overstimulation of the immune response. And this is a multifaceted system of resistance. It's regulatory T cells, it's myeloid-derived suppressor cells, it's a number of different factors. But one of the most important factors in the way that the immune system resists overstimulation is immune checkpoints. And so the immune checkpoint system, which you may have heard of, really is a built-in system in the immune response that avoids overstimulation. So it's really developed for a number of different reasons. One, of course, is to develop chronic stimulation of the T cell or antigen-presenting cell system. And so chronic stimulation leads to a variety of, the expression of a variety of inhibitory receptors on the T cell surface. These inhibitory receptors essentially are ITIM-containing receptors that dephosphorylate key signaling molecules within the T cell, and this leads to functional exhaustion of the T cell. So these cells are non-responsive to secondary therapy, they're terrible killers, and it doesn't really matter if they're tumor-specific or not, they're just not going anywhere if these receptors are actually engaged. And so I think that this, due to the immune selective pressure, many cancers express the ligands for these receptors and thereby create an environment that is quite inhibitory to immune function. So they essentially actively suppress these T cell responses. And so this has led to the development of a number of therapeutics that will target this axis. So if we understand how T cells become exhausted by the tumor, can we develop therapies that can block this exhaustion? And this is something that has really hit the immunotherapy field like a bomb and really has been a major focus for all the pharmaceutical companies out there and a number of different biotech companies as well. And so the two first molecules that really hit the market, the first was CTLA-4 work from Jim Allison's group and with IDEC and Medarex and other companies to develop molecules that will block this interaction. Another is this anti-PD-1, PD-L1 axis. And so there's a difference between these. They kind of act at a different phase of the immune response. CTLA-4 acts at the priming phase, and so this is when the T cells are first being taught by the dendritic cell what the neoantigen in the tumor is. There's a strong signal that leads to the upregulation of CTLA-4 and blocks this co-stimulation signal by competition. And so basically the ability to generate anti-tumor T cells is limited by the CTLA-4 interaction. Now in the effector phase, you have the role of PD-1 and PD-L1. And so in this phase, what we're seeing is that the tumor itself can, or tolerogenic myeloid cells, can upregulate the ligand of PD-1, which can then lead to functional exhaustion. So both of these things working at different areas really limit the anti-tumor immune response, and monoclonal antibodies against them have been shown to be extremely effective in propagating anti-tumor T cell responses. And this is seen here in this Nature paper looking at this lung cancer here in these patients. You see pretreatment of wide variety of metastases that are eliminated after T cell PD-1 checkpoint inhibition. And this is coincident with a massive infiltration of CD8 T cells within the tumor itself. And when you actually do the tumor biopsies, you can look at gene expression patterns. The gene expression patterns are predictively enriched for cytolytic T cell molecules like granzyme A, granzyme B, all infiltrating that tumor. These reinvigorated T cells are in there, and they're killing like crazy. So this did not go without notice. There's a variety of companies that have developed reagents targeting the PD-1, PD-L1 axis, both the first pembrolizumab from Merck on trial, nivolumab by Bristol Myers Squibb against PD-L1, Yervoy against CTLA-4, all of them in clinical trials, all of them actually now in clinical practice for a number of different diseases. But this is really just the beginning. So these three are now available, and they're out there. There are other ones that are other molecules and antibodies to block that are coming onto the market. But you can see there's a wide variety of activating and inhibitory receptors in the immune system. All of them are legitimate targets for next-generation therapy. And actually, importantly, combinations of these are probably even going to be more effective. Combinations of inhibitory molecules or combinations of activating and inhibitory antibodies, both agonistic and antagonistic. So the combination therapy, both in clinical trials and as I show you here in this animal model data, which demonstrates this is a tough animal model tumor where CTLA-4 has some effect over control, anti-PD-1 has some effect, but the combination really wipes out the tumor. So, when you go back and retrospectively look at the data sets, either in animal models or actually really in the human data, and you ask which cancers are most responsive to checkpoint inhibition and anti-tumor immunotherapy through checkpoint inhibition. One of the things that really came out was that the most mutagenic tumors like melanoma and lung cancer, smoking-associated lung cancer, the ones with lots of mutations are the ones that respond best to checkpoint inhibition. And this makes sense, right? So if there are a lot of neoantigens that are present, then you can reinvigorate those T cells, and there's a greater chance that you can that you'll cross-prime T cell responses against it, and then you'll once preventing the exhaustion, you can get good clinical durable responses. And so this is really what people have seen. So, but it's not enough just to have lots of mutations. Very recent work from the Stanton and Casada lab has really identified how these neoantigens really do interact with the immune response, and what's a good neoantigen, what's a bad neoantigen, and something quite a fantastic science paper that came out very recently. What they were showing really is that there's a difference in the quality of neoantigens. So it's not enough just to induce genotoxic responses against the tumor and induce lots of mutations. You really want to have universally shared, identify those trunk mutations, those mutations that are early on the tree branch and therefore most universally shared throughout the tumor. These later branch mutations, and even the branch mutations you can induce with genotoxic therapy like cisplatin, this really actually serves to only distract the immune response, decoy many of these T cells away. So you really have very strong immune responses against the mutations, but they're only representing a very small fraction of tumor cells. And so the critical aspects of this are to understand what the trunk mutations are, these big ones. Try and stratify these patients. Try to induce immune responses against those trunk mutations and bulk that up, and then come in with PD-1 therapy or CTLA-4 or whatever. So one could envision a combination strategy that would include loaded dendritic cells that through exome sequencing you've identified these trunk mutations in combination in order to prime these immune responses, such as like a steering wheel when you're guiding the response specifically toward these trunk mutations using DC vaccines, and then maintaining and invigorating these with checkpoint inhibition. And so we're just tremendously excited to see Sergio and Charles' paper put into action in a therapeutic sense in the next 5 years, hopefully. So overall, when you look at the checkpoint inhibitor space, that really great, fantastic responses, with 20 and 30% responses, and as we begin to stratify our patient populations and perhaps get to combinations, these will go even higher. You see complete remission in a variety of patients. They respond, I mentioned, the response depends on the mutational burden. The more mutations you have, the better, and importantly, the more of these trunk mutation responses you get, the better you get. But one of the key aspects of this is that the mechanism of resistance is really unknown. So we don't know why some people respond and some people don't. And so we really have to get to the bottom of this. And this takes a lot of analysis of these clinical trials, deep immunophenotyping. But another approach that groups have taken is to say, well, is there something that can be universally done for everybody, and really targeting, if we can identify specific target mutations or tumor antigen on the surface of these cells, why can't we just go ahead, skip all of this stuff, and get the T cells to attack them? These are the effectors of the immune response. And so this is where T-cell immunotherapy comes in, so cell therapy. And so we've been heavily involved in cell therapy now in my group here in Singapore to go after virus-driven cancers in our sense. So in this case, we're, much like many cell therapy groups, isolating T cells, growing them up in culture to be antigen-specific, where we know what the antigen is, and then infusing them back into the patient so they can go after and destroy the tumor. So in the trial that I'll talk about now, this is a trial for head and neck cancer, nasopharyngeal carcinoma, so EBV-driven cancer of the nasopharynx. We grew up this is in collaboration with To-Han Chong, who's the deputy director of the National Cancer Center here. So what we've done is we grew up those EBV-specific T cells and then infused them into the patients. We first analyzed them. The T cells are not specific for one tumor antigen, or in this case, viral antigen. They're specific for a breadth of antigens, so it's very difficult for the tumor to escape. And post-infusion, we can get very good biodistribution in the patients, including biodistribution at the tumor site itself. So within 24 hours, we see, well, actually with 4 hours, you see migration into the posterior nasopharynx and accumulation of these T cells. So in summary, it was the most successful trial for nasopharyngeal carcinoma. It gave fantastic durable responses. So we've got now 4-year survival on trial, but the key to this is overall 2-year survival of 67%. It's better than any other trial that's out there for NPC, and it's extremely encouraging. We've now moved this into an international phase 3 clinical trial with 27 sites throughout Asia and the US. So we're anticipating hopefully some great responses on this study. So overall, the deep immunophenotyping led us to identify some actual mechanisms of action and resistance. So for mechanism of action, if you look at the T cells themselves, individuals with strong T cell responses against the LNP2 antigen seem to respond much better. Importantly as well, your systemic response to infusion also predicted whether or not you were going to survive. And that was, if we infused you with the T cells and you responded with this Th1 polarized response, as reflected by your plasma proteome, this was a very good prognostic marker. You were going to be okay. And there was also a negative signature associated with non-responses. This was associated not with a lack of T cell responses, and these individuals also had T cell responses, of course, because we were infusing 1 billion cells, but it was actually a reflexive negative pushback, and it was the cellular pushback, the expansion of activated regulatory T cells and the expansion of these myeloid-derived suppressor component. And so in those individuals that were not responding, we saw this real signature and actual expansion of these myeloid-derived suppressor populations. And so this really leads to obviously a rational redesign of the clinical trial in which we maybe in this short time period are able to eliminate these myeloid-derived suppressor cells and precondition you to be more receptive to immunotherapy. This is true for our therapy, maybe other therapies as well. Okay, so it was, you know, in the context of virus-specific cancers where the virus is driving the tumor, so for NPC that's the case, your antigens are there. You go after the virus antigens. But in the case of many tumors, these viruses are either not present, they're not part of the associated transformation event, or their viruses aren't there anymore. And so you need to identify something else. And so an approach that was taken was really to take these T cells and actually combine the power and specificity of monoclonal antibodies with the effector function of T cells. And so you may have heard of chimeric antigen or CAR T cell therapy, which uses these monoclonal antibody specificity, these the heavy and light chain variable domains, to bind to a particular tumor antigen, combining this in a genetically engineered construct that has a signaling domain of the T cell receptor. So one could engineer a T cell that was a fully potent killer, but instead of signaling to its T cell receptors, it uses this chimeric antigen receptor to destroy the tumor. Enormous, promise on paper, and actually enormous promise in practice too. We've seen this kind of surface binding and attack. So these chimeric recombinant antigen receptors are actually destroying the tumor itself. And so there are huge advantages to CAR therapy. One is that killing of the tumor cell is no longer MHC restricted. So any tumor antigen on the surface becomes your next target. It doesn't even have to be a protein antigen. You can go after glycolipid antigens, carbohydrate antigens, you name it. If you can make an antibody against it, you could potentially use it for potent tumor efficacy. Now, there are downsides to this. One is that this is not an inert T cell. The signaling and killing event is intimately linked to cell division. And so these T cells, as they kill, they begin to divide, and they're extremely potent with this very strong signaling domains that they put on them. And so this can lead to a potential for very high toxicity. These toxicities being addressed now, but we've seen, obviously, you've seen some tough times on some tough side effects on some of these trials. So this is just some example in the mouse system where different generations of chimeric antigen receptors can clear the mouse models in the mouse system, but also, more importantly, actually in humans themselves. And so this is work from our collaborators, Steve Gottschalk's lab at Baylor College of Medicine, looking at human sarcoma with a HER2/neu CAR. So this is an antibody against HER2/neu that has now a chimeric signaling domain, including the CD28 signaling domain and the zeta domain of CD3 receptor, T cell receptor. And you can see that pre-infusion in the PET scan, you can see this tumoral mass. Post-infusion, 6 weeks post-infusion, you can see in the immunohistochemistry panel, massive necrotic tissue and an elimination of that tumor. And this is true for a number of different tumors that people have tried, particularly in the lymphoma space. So what we defined is remarkably certain responses. I mean, responses that we never saw in our DC Vax trials, 90% complete responses in the context of B-cell lymphomas. So the extreme potency, as I mentioned, has led to problems with toxicity. And I think more importantly, the associated, there's been limited efficacy so far in the solid tumor space, but everyone who's in this field is trying to work on this problem to try to understand tumor penetrance. So, okay. It's not just T cells. There's some fantastic groups that are trying to modify NK cells. They have some advantages in terms of multiple mechanisms of killing. Work from Dario Campagna's group and Charles Sentman's group and others that have pioneered this NK cell CAR field. There's enormous potential for this as well. And so we're looking forward to more clinical trials in this space. Okay, so this looks like a little bit of a glitch issue, but I think you can take the message home. So as much as we can bring in things like the dendritic cells to prime new immune responses, the checkpoints that cause these T cells to become not exhausted, we can also envision a situation where maybe we'll be able to overcome these many of these things with checkpoint, with genetically engineered T cells, with dendritic cells to prime the breadth of response. But I think there are a huge number of hurdles that are still in front of us. And one of the biggest hurdles is really this tumor microenvironment. So the metabolism of this environment, the acidic microenvironment, the pH, the glycolytic polarization, where it basically eats all of the sugar up so the T cells can't function, are really deleterious to inducing anti-tumor immunity. In addition, the metabolic microenvironment actually polarizes myeloid cells to become more tolerogenic. And this tolerance it's not passive. It's not just a lack of T cell responses. It really is dominant immune tolerance. And so they actively prevent and polarize and exhaust these T cells. And so this is really the next step in where we need to go. So where are we in terms of the next frontier? How do we overcome this dominant tumor microenvironment? So just to finish now on the last two slides, I think that we have resurrected Claude Bernard and his understanding of the immune response. I think the self-non-self really does serve us well in developing new therapy for immunotherapy. And I think to finish on my title, and because we're in Asia, I think that, a quote from The Art of War itself. So if you know your enemy and know yourself, you need not fear the result of 100 battles. So you really need to understand this concept of not self, non-self, how to manipulate it, and then apply that then to breaking down these natural barriers to induce strong tumor immunotherapy. So just as a reminder, technology drives science, science drives medicine, and that innovation really does occur at this interface. And so I want to thank everybody for their time. And I think there's a couple of minutes for questions, but I want to make sure that I thank everybody that helped with this work. I think it reflected decades of science and research, particularly Han Chong, Jacques, Joe, Carolina, and Steve and Malcolm Brenner at Baylor College of Medicine. So thank you guys very much. And I think the moderator will take some questions, and I'm happy to answer them.
Thank you, Dr. Connolly. Great presentation, very important information. Questions can be asked by clicking on the green Q&A button, typing your question into the Q&A box. We'll get right to it since we have several. Our first question, how do macrophages differ from dendritic cells?
Okay, thanks. It's a spectacular. Thank you. It's an absolutely fantastic question, and it's an area of tremendously active research from many of my colleagues. But to put it simply, dendritic cells are specialized for antigen presentation and T cell priming. So that is, they can take a naive T cell that is totally unpolarized and expand it and polarize it in a particular direction. They also have a unique property in that they, in their immature state, they don't have acidified endosomes, so they don't digest. They basically don't digest the proteins until they get a non-self signal. And so this allows them to capture antigens in the periphery, travel to the lymph node and present, whereas the macrophage is much less capable of that. They constantly digest all the time, and their priming capability isn't quite as good.
Great, getting right to our next question. How are dendritic cells regulated?
Sure. So I guess I think there's a couple of ways you can interpret that question. One is the developmental regulation of dendritic cells. How do you get more of them in the periphery? And there are common dendritic cell precursors that have been identified in bone marrow development. Most of the dendritic cells that we think about in the periphery are derived from those precursors. And once they're in the periphery, they can then, their activation is regulated by pattern recognition receptor engagement. And so naturally, the dendritic cells will be normally tolerogenic. That is, when they stimulate a T cell and they present self-antigen to the T cell, if the T cell recognizes it, in order to become activated, it needs a second signal. And so that second signal is called co-stimulation. And in order to polarize it into third signal, which is derived from cytokines and receptors. So naturally, self-antigens presented without these sort of cytokine signals or co-stimulation signals, which are coming from pattern recognition. So they would tolerize those T cells or energize those T cells. So it's regulation, it's dynamic regulation between immature and mature state is through cell signaling through pattern recognition receptors and inflammatory cytokine receptors.
Great, thank you. I think we just have time for one last question. Is it possible to activate the immune system too much and produce autoimmune disease?
So that's a absolutely wonderful question. So we have seen that in some cases in our melanoma trials, where a side effect of the therapy is the induction of vitiligo, where you begin to kill the normal melanocytes. Obviously, in late-stage cancer, this is seen as a very good thing. If you're killing those melanocytes, you're really killing a number of these. You'll be killing the tumor cells with even greater efficiency. However, this is something as we move forward with more effective immunotherapy, things like the CAR Ts, for instance, or even in the context of combination checkpoint, activating and inhibitory blockade. We really have to keep an eye on this more. So in general, this is not something that we've seen enormous, it's not been a huge problem on trials, but as we become more and more aggressive with immunotherapy, we need to keep an eye on these kind of side effects.
Thank you, John. Wonderful presentation. Lots of great questions from our audience. We are out of time. I would like to let our audience know that this presentation is going to be available on demand through November of 2016. You'll be receiving an email from LabRoots alerting you as to when that is available for replay. I want to thank Dr. John Connolly again for his time, as well as our sponsor today, Affymetrix, part of Thermo Fisher Scientific, for making this webcast possible. If we didn't get to your question today, we'll definitely try to follow up with you. Thank you for joining us today, and we hope to see you again. Goodbye.